Okay, thank you so much, Michelle. Good morning, good afternoon, everyone. This is a webinar on safety organized by the ASA Biopharm. It will focus on safety monitoring in clinical trials. I am Rima Izem. I will be both organizing a little bit and then also the moderator of this webinar. So the agenda in the next slide is shown in the next slide for this meeting. Yes. So first I'll give some welcome motivation also for why we have this session. And then the bulk of this webinar will be several invited speakers who will give very short presentations on their methodology. Then we'll have hopefully prepared discussion questions and maybe some opportunities. If you could please mute your line when you're not speaking. Thank you. And then finally, some closing and next steps. After the welcome and before some of the invited speakers, we'll have a short poll as well so that our speakers know a little bit more about you who are in the audience. Next slide. So who's organizing this? It's primarily the ASA Biopharm Safety Working Group, more specifically Workstream 2. I would like to acknowledge a contribution of many people from the Biopharm Safety Working Group, including the communication and outreach, and the lead of the Workstream 2, Michael and Melvin. Also would like to acknowledge other organizations that also discuss safety, and that is FUSE and PSI. On this slide, you will see links to all of these professional working groups, and many of the presenters today are part of at least one of these working groups. Next slide, please. So who are our speakers? We sort of did a review of the literature, especially published tools and published methodologies. And here are some of the names that came out. This webinar will not be completely representative of the whole landscape of methodologies that was developed in safety, but I hope it will give you a flavor of all of the work that's being done and also where are some of the similarities and the challenges. I'll be moderating this webinar, mostly trying to make sure that everyone has the time to discuss their slides, and Barbara Henriksen will take the lead in the discussion portion of the webinar. Next slide, please. So why did we come up with this webinar in this format? Safety monitoring is very important. Several regulatory guidance documents recommend to do it judiciously. That is not the one-size-fits-all, but rather really thinking about the problem and how to approach it on a case-by-case basis, depending on what type of program you have and also what type of study you have and maybe what type of the seriousness of the adverse events. They recommend doing it with aggregate reports, and this is where the quantitative aspects of how to do it comes into play, and you will see different ways or different approaches that the speakers have taken to address this problem. So what are the learning objectives today? Mostly to get an overview of the existing methods and tools, to identify what are the common strengths and barriers, and to identify the next steps in dissemination or implementation. More particularly, we hope that this very short presentation is only the first webinar of many to come in the next few years, where we can delve more deeply on each one of these methods, or maybe some possible extensions. So that is why we're doing it in a landscape format, where we are not focusing on one methodologies or two speakers, for example, but multiple speakers to really give the lay of the land. We're hoping also that this webinar itself can serve as a reference and be on our websites to maybe get you started in the use of some of these methods, or at least identify who to talk to if you need some help. We want the panel discussion to focus on common challenges, which is implementation, because that's been our experience, is that there isn't much dearth of methods, but rather that implementation has become the biggest challenge. We'll finish also with a call for action to help us identify and maybe prioritize which topics to delve on more for, again, future. Michelle, have we heard from our attendees?
Yes. So I'm going to end for now.
All right. Well, I guess we were successful in advertising this within at least the pharma industry. That's the majority of you who could join us today. Thank you for coming. And we have regulators as well and some academic attendees. And in terms of experience, many of you are already experienced in use of these methods. So I hope you can join us in the discussion and also maybe putting questions or trying to shape future presentations. Thank you. Go ahead. Greg.
So the first blinded safety monitoring procedure was published in 2011. Next slide, please. It was a very basic idea, so it's easy to implement. There's a very small barrier to entry here. It assumes binomial data and uses a conjugate beta prior. And you can calculate probabilities that the pooled rate is greater than some critical rate or some reference rate given the data that calculate the probability that it crosses some thresholds. Next slide, please. Now, interestingly, this came out before or after the IND safety reporting final rule came out, but I was not aware of it at the time. But I would say that the forces that led to the final rule for the FDA are the same forces that led me and others to develop these blinded safety monitoring procedures. The FDA refers to it as the trigger method in their final rule. Next slide, please. So Patrick Schnell and I wrote twin papers. I'm going to talk about the more conceptual one. This was really about the procedure and for leveraging the scientific expertise and medical judgment of multidisciplinary safety teams. And there's three bridges really, which bring the clinical and statistical scientists together so that they can communicate and understand the questions, answer the questions, provide useful evidence for decision making. So the first bridge is coming up with the background rates. Those background rates inform the model parameters. So the second bridge is taking those model parameters, running some simulations, coming up with operating characteristics, make sure that it can properly work. If you don't have good enough estimate on the background rate and there's not enough information coming in, enough events coming in, then these blinded safety monitoring procedures aren't going to work very well. And the last bridge is communicating the results. Next slide, please. So you get something like this. You can calculate the probability threshold boundaries. And notice there's not just one threshold, which would be like a decision rule or a p-value, right? We're trying to measure the data. Go ahead and click one forward. And so the colorful lines are the probabilities of relative risk elevation. The red is 90%, blue is 80%, green is 70%. The solid line is some reference rate, critical rate, whatever you want to call it. And the dotted line at the bottom is the background rate. And so you can see over time where you fit in here. And, you know, right now maybe getting a little bit perilous, right? The team might be on edge. Click one forward, please. But over time, you see it's coming down here. So this might provide sufficient evidence for the team initially to look more frequently, but as time goes on, that it looks like it's coming into a comfortable place. Next click, please. And then by the end of the trial, it's designed to look scary in the beginning and cool down. But this provides the evidence that's available in the blinded data for the multidisciplinary safety management team without unblinding them. Next slide, please. Patrick?
Yep, thank you. So the next few slides here are based on the previously shown paper and this one, and get just a little more into the statistical mechanics. Next slide. So the general idea behind the statistical analysis, we have two arms in the trial, control and active, and they're allocated in a known ratio, say 1:1. But since we're blinded, we of course only observe events in the combined sample. If we know the control arm rate, say from historical data, and we observe some event rate in the combined sample, then we can solve for the event rate in the active arm, right? So alternatively, if we set a maximum allowable rate in the active arm, that fixes the maximum allowable rate in the combined sample as well. Next slide. So the Bayesian framework allows us to relax that a little bit so that we don't need to have a known control rate and a completely unknown active arm rate, but instead just strong prior beliefs about the control arm rate and weak prior beliefs about the active arm rate. And when we make observations in the combined sample, they will naturally influence our beliefs about the active arm where we had weak prior beliefs more than our beliefs about the control arm, for which we had strong prior beliefs. Next slide. And we've applied this strategy in two settings where the type of adverse event that we're looking at determines which type of model we use for planning and analysis. So if events happen early and at most once, then we can use a binary outcome model. But if it happens dependent on exposure time and can happen repeatedly, we can use a Poisson model instead. But we should both think about these assumptions at the design stage and check them in the analysis stage, which we can do even while blinded. Next slide, and Greg will take over again.
And I just wanted to put a quick plug in here for Emily Gorin. She did some amazing work. I think this is the only paper that is specifically designed for a meta-analytic approach. Next slide, please. And it takes into account that there could be some heterogeneity in the different studies. So you can use homogeneous priors or heterogeneous priors, and you can do sensitivity analyses where you take one or more studies out of the analysis. Next slide, please. And then you can come up with estimates for each study. And
thank you, Greg.
Yeah.
We'll have to move to the next speaker. Sorry to interrupt.
No, that's good. Thank you.
Yeah, good. It was perfect timing. Okay. Our next speaker is Saurabh and Brian, and they will also be co-presenting.
Thank you, Rima. Thank you, the organizers. So BDRIBS stands for Bayesian method of detecting potential safety risk using inference on blinded safety data. Now, as you can see in this plot, it's like giving you a planning of entire program of how and when you would like to monitor and when you would raise a signal. And if you can plan everything ahead of time based on what you want to monitor and how. We are going to talk about a little bit about the method just briefly, and then Brian will talk about the implementation method part of it. So can we go to the next slide? Yeah, so randomized clinical trials are typically conducted in a blinded manner during development of a new therapy, and ensuring patient safety in this trials are extremely important, and for that we need to have a proper safety surveillance, as other speakers have alluded to. It is important for both regulators and sponsors to find a quantitatively rigorous method to carry out this task. And the BDRIBS offers a very unique way of quantitatively, rigorously monitoring and identifying any potential safety risk from this blinded data. And it does so using aggregated data, and also it uses a background risk or background rate, what is obtained from historical data for the event in the control. Can we go to the next slide, please? So it starts with the historical data, and then it models the relative risk of having a signal, having an event in the investigator new treatment group versus the control group, and then it computes the posterior probability given the blinded aggregate data. So we can always determine that what is the how much relative risk we can allow in that particular program, and then monitor the posterior probability as we have aggregated data available over time. We can do that for one single study, or we can actually plan for multiple studies to be monitored together. And once the posterior probability, or we can even use the base factor, which gives a natural scaling of where the thresholds should be drawn. We can use either of this in order to see whether there is a trend or safety signal. Then we can take several next steps. And that next steps can range from a group of experts getting together to discuss the cases and communicating in concern to the DMC or to unblind the clinical trials. And it does so on an ongoing basis, as I pointed out before. And we have actually, Brian, I, and other co-authors have put together two papers on that. The first one is the methodological paper, came out in 2018, 17-18 timeframe, and then implementation paper was talked about how different scenarios can be tackled, and there is a Shiny app that comes with it. Brian will discuss more about those implementation steps. Brian, maybe you can take over.
Sounds good. Next slide, please. So Saurabh mentioned, right, so BDRIBS is modeling the relative risk. So we're trying to answer questions on, such as how likely is the rate of the event for the investigational drug twice that of the control. Now, of course, we're in a blinded detection realm. So we really have a lot of uncertainty. And two things that you might ask yourself about, like how good are my assumptions about the historical event rate? Or how likely is a blinded event happening in the investigational drug? These questions can be directly investigated via sensitivity around the priors used in the BDRIBS model. We can also use interactive graphics to look at different what-if scenarios concerning the observed data. So we developed Shiny BDRIBS, a suite of graphics that may be useful. Next slide, please. So I just, in the interest of time, I just wanted to show you what Shiny BDRIBS has to offer. It opens to an instructions page, which gives you a basis for understanding what we're trying to do, with the prior assumptions being able to be put in by the user. Next slide. And we can look at directly at the posterior graph based off of the observed number of events and exposure. Next slide, please. And we can produce a contour plot, which is similar to what Greg Ball showed earlier and what we had in our starting slide. We can set up the parameters as the situation demands. Next slide. We can investigate a different number of events and how that impacts our probability of exceeding our threshold. Next slide. And look over time at the different what we've observed over time, looking at the range for the different observations that we've had. Next slide. And investigate the range of possible probabilities. How good is my 75% greater than the threshold by looking at the historical event rate variability. Next slide.
Ryan, your time is up. If you could wrap
so
up.
This is it. The implementation guide is also available to you. And the next slide is the summary. Thank you very much.
Thank you so much. The Shiny app is definitely very, very useful, and thank you so much for sharing the papers. Our next speaker, is Nils Penard. Go ahead.
Thank you, Rima. Today, I'm going to introduce a Bayesian method geared toward the detection of safety signal in ongoing blinded trials. Events of interest could be serious drug reaction or anticipated event safety topics of interest. This method has been formally described into a paper our group published in 2023. Next slide, please. Very short outline. I will first go over the novelty aspects, then present a few relevant references, and we'll conclude with barriers to implementation. Next slide, please. So we'll first describe the two main categories of data. The first category is the ongoing study data. As shown on the left, it could be a blinded ongoing trial, or as shown on the right, could be an unblinded ongoing trial. Next slide, please. The second main category of data is historical studies data. Those data mostly originate from completed clinical trials, but also could come from the literature or from large claim databases. The method presented here is flexible and can accommodate for either patient level or summary level, or patient level and summary level, although some caveats apply in this. Now, those historical studies will be used to build our priors, summarizing our knowledge regarding the events we wish to monitor. Next slide, please. And here is a symbolic depiction on how the priors are derived. On the left side of the illustration, you see a set of carefully selected, completed ongoing trials with different characteristics. Then we apply a meta-analytic predictive prior Bayesian method, which will be used to compute the background rates of the event we wish to investigate. The outcome of this step will be an empirical distribution shown here in the middle. And the next step will be to build an analytical representation of this distribution. To do so, we fit a finite beta mixture. This offers a better, more accurate, more flexible way of fitting the distribution rather than using a single beta. On the right side of the screen, you see an example of such beta mixture with two components. You will observe that each component is a single beta function with shape parameters A and B, and each component has a weight, and those weights add up to 1. And this is interesting for working in the probability world. Next slide, please.
Sorry?
The internet don't work?
No.
Sorry. Okay, now that we have our priors, we can feed them together with the data originating from the ongoing study to our second Bayesian model. This model, in the case where a treatment location is not known, this model will assume that each treatment can potentially be allocated to either the active or the placebo group, and all of this will be weighted according to the randomization ratio. The statistic of primary interest resulting out of this step is shown here, and is the probability that the true rate in the active group is larger than the true rate in the placebo group, knowing what we have observed in the ongoing study, blinded study. Next slide, please. Here are a few relevant references. It's just a selection. On the top of the page, you'll see our paper. Now, jumping on the software front, at the bottom of the page, on the left, at UCP, we have implemented this methodology using an R package. Unfortunately, this package is not available at the moment, but we are working hard to make it available. In the meantime, you can pay a visit to OpenStatsWare website. They are a special interest group, ASA, special interest group, and 3 of their members, namely Daniel Sabonis, Christian Brock, who's also first author on our paper, and Doug Kalkoff, have implemented the methodology as a Julia package. Next slide, please. So this is the last bit of information I wanted to share with you, the barriers to implementation. The first 2 items here are a rule of thumb with use at UCB, and they are the result of simulations we conducted with a methodology. And that is that we only apply this method when we can observe at least 3 events in the ongoing study, and if we have at least 50 effective patients enrolled in the ongoing study. Other barriers to implementation could come from prior data conflicts. So it might be that you have a compounded factor in the priors that you haven't seen or thought of. It could be that the target population is ill-defined. More complex design are not covered by the method. We are stuck with active versus control, and we cannot, for instance, manage different doses. And finally, another barrier to implementation, and again, all of these barriers are not specific to this method or even to Bayesian method in general. You can be facing a dearth of data. So active arm data is hard to come by, and the indication and the population you are studying might be rare or hard to find. Just to conclude, I wanted to thank many of my colleagues in the little blue box at the right side of the screen, and I apologize for the ones I've not been able to put in that little box. So all these colleagues worked on the package or on the paper and other analysis during the year. That's it. If you have any question or if you wish to discuss this further, please do not hesitate to contact me. Thank you.
Thank you so much, Nils. So without further ado, Dooti and Stefan will be talking about other methods and also software available for visualization. Go ahead, Dooti.
Thank you, Rima. I also want to start with a big thank you for this group organizing this, Susan, Rima, and others, giving the opportunity for us to come and talk to you as a community. Yeah, this is the presentation is going to be focused on what Boehringer is internally doing for the topic of safety, both in terms of visualization and also analysis. So Stefan and I are both very happy to be able to do this presentation with you. Next slide, please. Standard disclaimer, please, let's go, sake of time. Thank you. So as you can see also from today's presentation, we don't really have a dearth of like new approaches, how to explore and analyze and draw inferences from safety data, and more on the way of even becoming a reality. However, at least what we have internally observed, that this topic is also a scalability issue. Which basically means safety is something that all trials at all phases during development needs to consider. And of course, we want our trials to be able to take advantage of these methodological advancements and the technology that's available now. However, it's really difficult to jump into ideation to implementation in a quick time. And therefore, that's the little bit of angle the presentation will try to explore today. We are trying to leverage an open source modular framework so that we can enable quick adoption and broad application of these methods. Next slide, please. Stefan?
Before jumping into the safety-related aspects of our presentation, we want to make sure that the audience has a superficial understanding for the modular approach to our Shiny-based app building is. Imagine a set of Lego blocks. Dependent on the blocks available, you might be able to build very different things like Lego motorcycles or cars. With common building blocks like, for example, wheels or a 3x2 building block. Similarly, a modular approach, in this case, our Shiny-based, is a technical programming framework, and instead of Lego blocks, there are functional building blocks that we call modules to easily and quickly build modular Shiny apps according to your trial or project specific needs, for example, to support your exploratory safety analysis. Today, we quickly demo two modular frameworks, DaVinci and Teal, which are both R Shiny based and open source. Here's an overview of the modules that we have already available within the DaVinci module framework. And those highlighted light blue are modules developed in particular with the safety use case in mind, like a eDish plot module or patient profile module. There are those modules highlighted in gray that are more generic but might also be very useful to support your safety review analysis. So now we prepared a short video with a dummy app to demonstrate how DaVinci could support exploratory safety analysis. On the left side, you see the data loader to switch between data sources, for example, trial project pooled data. And below it is a data filter where you can filter outputs according to dataset variables, for example, age. On the top side, there's a navigation panel that lets you switch between different output modules. Now we entered the clinical timelines module. And here you have an example of interactivity. You go over a data point that gives you more information. Now we switch to the lab line plot, where it shows mean of all lab values over time grouped by treatment arm. And now we switch to the subject level, and you can drill down on a particular data point and jump over to the patient profile of the respective patient. In there, you can further drill down on the different data points, lab values in this case. And finally, we go over to the eDish module, which can give you a patient with hepatic failures. And with that, we conclude the demo.
Also wanted to introduce a little bit more complex application of this tool. This is the BSafe. And we just heard a couple of speakers already introduce the Bayesian map prior approach. We also know that the map prior, the meta-analytic prior approach is broadly applicable now in efficacy endpoints, but of course you can do so for the safety. And the BSafe is an answer to that. The good news is that the BSafe module is part of Roche's Teal app gallery. So we wanted to explore if Boehringer can provide a module to another framework, and this is also what happened. I can go to the next slide, please, and quickly show you very few, Rima, just a few seconds to show how the app looks like. So, yep, so you get started. You can upload your data. As you see, this will all happen in a matter of seconds. You select the patients you want to look at. It gives you both exposure adjusted and also incidence proportions. Select the safety topic of interest. Calculate your map priors. You can control the heterogeneity you're expecting in those trials. If you're unsure about the prior data conflict, you can use a robust map prior piece. You enter the new trial information, and then you can make decisions based on this updated data. Very quickly, a few seconds, you can choose the different comparisons that you may want to run, which we very typically observe in safety analysis, right? You want to compare different arms. And as you can see, the simulation will finish in a few seconds. And here is your typical looking adverse event summary tables that's then available for download. I will stop the thank you very much. Just to wrap up, again, the problem of what we do in safety is also an aspect of scalability topic. The implementation piece has a lot to think on, and if you are interested in further discussion, please feel free to reach out to me or Stefan. Thanks a lot.
No, thank you so much, Dooti. And one more pause. We'll go to the next speaker, Lian Lin. Thank you.
Hello. Yes. So today I will talk about the two topics. The first topic is about the meta-analysis of blinded and unblinded studies. So next slide, please. So just a few background for why we come out with this approach. So as the clinical development program evolve, typically you will saw both the blinded data, which is for ongoing studies, and unblinded data for complete studies. So how we can combine both source data together to understand the evolving safety and also calculate the risk. So that could be our questions. So we proposed two meta-analytic approach to synthesize both form of data and also how do we can use that for decision making. Next slide, please. So the first method is really obvious. So we don't have to meta-analysis both source data together. We can just present them separately, side by side. So there are a few rationales. The first is that those two types of data source representing different understanding of the data. So complete studies from the past and ongoing is represented from the current data and involving. And also it can have a complementary strengths. Unblinded study can offer direct comparisons. However, the blinded studies that is just blinded, so they not support the direct comparisons. But we only can use the background rate to make the comparison and the inference. Next slide, please. So I will just briefly go through the method, but for time's sake, let's go to next slide. So the second method is combine those blinded and unblinded data together in a meta-analytic approach. So here in this paper, we use the power prior. So the blinded study and unblinded data can be formed together as a likelihood, and then we can estimate the posterior estimations, and we can use the weight to weight how much we can leverage blinded and first unblinded data, and in the finally we can use different weight to see how the data involving use a different weight. Next slide, please. So for barrier to implement this method, sometimes it's difficult to get a sensible amount of information to be combined. So in that sense, we could use a different weight and to examine how the sensitive that is, and present all the data together and making multidisciplinary decisions. So here is a reference, including the paper that are only using the blinded data, and also the paper they combine both sources of data together. Next slide, please. So this is another topic about bridging blinded and unblinded analysis. So we all know that we have the FDA guidance, especially in 2021. So officially they adopt the trigger approach for blinded aggregate analysis. So the question is, what is the statistical properties, and that we conduct the last analysis sequentially. So here we propose a two-stage framework that including the blinded analysis to flag potential safety signals and refer back to the DSMB or other unblinded body to locate the data more quantitatively, more quantify the association. So in latter sequential ways. Next slide, please. So this is the diagram. So we have first stage and second stage. So first stage, we look at all the safety event and using the blinded analysis to flag the potential adverse reactions. But also together with the other safety events were documented in such as IB, DITS, or IMPs, that including the anticipated event or the expected event. All those events can package together to refer to for the unblinded analysis. Next slide, please. So, so this is just some methodology. Basically, is we are using the Bayesian method, and unblinded analysis. We are looking at the false discovery rate, and so if you look at the next slide, and this procedure can help us to have a more strong control for false discovery rate among the multiple safety endpoints, but also increase the powers. In addition, this paper help us provide some statistical background how we can look at the data in these sequential ways. So this reference you can take a look after this webinar. Thank you.
Thank you so much, Lian. So our last but not least speaker is Shihua Wen from Novartis. Thank you, Shihua. Go ahead.
Yeah, thanks, Rima. Hello, everyone. And I think my brief presentation today is based on our TIRS publication, named as Quantitative Safety Monitoring in Clinical Trials, the application of multiple statistical methods. Only have one slide for this presentation. This paper was published in 2020. However, I think the majority of work was really done in 2015 or 2016 time. And this is a joint work for me together with Dr. Jiabu Ye, Dr. John Schoenfeld, and also Dr. Saeed Islam. And also I want to really appreciate Dr. Saeed Islam. He provided actually a lot of comments and encouragement for this work. But unfortunately, he actually passed away during the COVID time a few years ago. And also, besides this publication, I also actually put two additional reference for this on this slide. And I think especially for this one and other paper, the 2018 paper, I think that's really a very nice paper and summarize majority of the quantitative methods used for clinical safety monitoring and reporting. I think in our paper, we describe actually about three methods in our paper. I think the methods we described in this paper are not really new, even at that time. And also, I think this is not only just for infrequent events, it could be general safety event as well. The first method, we applied is actually called the wild SPRT method, which is the sequential probability ratio test. This is obviously not a new method. It could be actually traced back in 1940s, and this is actually a frequency method. The method actually was probably described in the previous presentation. You have seen, and this SPRT method is fairly probably, I will say, straightforward to be understood by different stakeholders. And what you need to specify is just a few parameters like alpha, beta, which is type 1, type 2 error, and also the null hypothesis, which is something you can consider as like a background rate P0, And also you need to specify your alternative hypothesis as well, like P1. And with these 4 numbers, you can actually set up your safety boundaries. And if you observe the number of events is actually across this safety boundaries determined by those parameters you set up, you will claim you actually detect a safety signal. And in addition, I think one merit of this SPRT method is actually it automatically adjusts the multiple testing due to this continuous monitoring. And the next method we described in this paper is called the maximized sequential probability ratio test, the MaxSPRT method, which was originally developed by Kudov, which was actually used in the post-marketing surveillance for some vaccine on their safeties. And of course, this method can be used both in the post-marketing setting and also in the clinical trial setting as well. And I think one merit of this MaxSPRT method compared with the regular SPRT method is that you don't need to specify the P1, but it actually automatically takes care of a composite of the P1, which is the alternative hypothesis here. And the third method we described is the Bayesian framework. And in particular, I want to say this Bayesian framework is based on really like the methodology proposed by Peter Sohl and also Simon in 1994 papers. And I have to also give credit to Greg Ball, who actually introduced this method to us at that time. And obviously, this is Bayesian method really provide a unified framework for continuous safety monitoring. And also they use probability statements, try to set up the boundaries, which you have already saw in the previous slides. And also, I think this is also an advantage of Bayesian method. Why people love it so much is because they can incorporate the prior, the historical information in the methods. And also, you can have many ways to actually try to tune the priors, either like downweight or upweighted, or even dynamically try to borrow the information from the historic information. Also, I want to point out that all the 3 methods we are talking about here in this particular manuscript is only based on particular AE. So we are not actually taking into account of the relationship between the safety signals or the AEs. And there are some other methods, for example, in Bayesian method as well, like I think based on the Berry 2004 paper, they talk about the hierarchy structure to take care of actually the relationship between the AEs. We also actually have a Shining app at that time developed it for these 3 methods, but we did not continually develop it. So I believe there are already actually much better Shining app for the quantitative safety monitoring. Thank you.
Thank you very much, Shihua, and thank you to everyone. Really, this was a very hard exercise to describe sometimes several references or work over several years into just a short 5 minutes. So thank you so much to everyone who presented here. As I said early on, you can find the bios for every speaker in this session today in the handout for the presentation. This was the flyer, and it will be also available with the videos so that you can have references, but also the names and the contact information for the people who are presenting today. We couldn't invite everyone who's worked in safety and safety monitoring. So here are some references that some of the speakers today already indicated for related work. Some for clinical trial, but also some that was developed in post-market safety monitoring that is still relevant, including some of the ones that was in Shihua's slide, MaxSPRT and wild methods. I think that's it we have for at least the presentation portion of the webinar, and I give it to Barbara for the discussion section.
Thank you very much, Rima. This was very interesting, and I'm coming at it as a clinical safety scientist, so not as a statistician, but I just emphasize how important it is for the safety scientists to collaborate with their statistical colleagues in trying to implement these methodologies in the appropriate situation, and for the statisticians to help the clinicians understand the data that's generated. So the first question we had was, how do you advocate for the important role of quantitative methods of aggregate analyses and safety, and how do you prioritize their use? And I'll just say very quickly, in my own experience, that it's very important to have a champion at your particular company in regards to advocating for the use of some of these methodologies. And I personally found them the most helpful for larger studies that are going to generate a lot of serious adverse events in particular, or that could be smaller studies potentially for oncology as well. But I think those are the studies where you're going to see multiple events of a like nature, that you really need to do this more quantitative assessment for any events that are going to be occurring in the background of your patient population. So individual adverse event review alone is really insufficient to judge causal association with your study drug. So maybe anybody who would like to further comment, if you have your hand up, that would be great. I can call on you. Anybody who would like to add? Dooti?
I just want to get a start on this, and maybe others can also join in and share their experience. This especially happened to us when we were working on both the tools that I presented to you today in this presentation. While really thinking about the inception of those tools, we had our so-called champions from our safety group. And they have been invaluable, both in terms of setting the right strategy direction and also giving feedbacks on a regular basis, so that we know, are we really addressing a pain point that they have, or are we just innovating in silo? And this approach really also helps in creating that adoption mindset, right? They own the product just as much as we do. And therefore, that really creates that leverage for them to say, oh, yeah, this is also something I helped build, and therefore I would like to go and talk to other colleagues in safety perhaps and talk about it. So we have taken that approach within the organization a lot. When we present, we have our safety colleague actually present these tools to the safety colleagues. Always goes down as well appreciated, and a better approach than just in silo, one organization talks to the other one. I just wanted to highlight that example.
Yeah, thank you very much. Brian.
Yeah, I want to echo what Dooti said here, right? I think the partnership between the two organizations is essential, right? So that we are providing information that is relevant to the role, that we are getting feedback on how we communicate that information in a way that makes medical sense, makes sense to, our role of quantifying things is actually a very powerful thing, because it pushes decision making one way or another. So, making sure that we're communicating not just what the quantity is, but what are the assumptions for the quantity? What are the different things for us to be concerned about is an important part. Advocacy, as you mentioned, Barbara, right, requires to have some champions within the organization. Without someone believing in the things that you're doing, it is incredibly hard to get a methodology to be implemented.
Yeah, thank you. Yeah, and I think prioritizing use is helpful because sometimes if you want to convince people to use these methodologies for all studies, that's a little daunting to them. So you have to really, I think, focus in on those programs that can most benefit from this, and maybe even start doing pilot projects first to demonstrate benefit, and to help convince senior management or others of the value of these approaches. Saurabh, you have your hand up?
Thanks, Barbara. Yes, I just wanted to follow up on the points Brian, you and Dooti are making, is that the planning is extremely important in this area, and how we tackle and when we will have the aggregated data available. Those things are important, and methods like BDRIBS provides that kind of provision. Especially, we'll be happy to come back to this platform to discuss more in detail that how you can plan ahead and what are the things that you can explore, the sensitivity. All these things are very important, and this to be developed in a partnership with the safety team and statisticians, clinicians, and can be developed program-wide. It doesn't have to be for one study. It can be done for the multiple studies.
Yeah, and I think the more we can do, as much as we're able to communicate examples of where using these methodologies made a difference in programs. I know sometimes proprietary information or considerations come into the way of doing that, but as much as we could do that, I think that would be also very helpful. Greg?
Yeah, I just wanted to double down on something you said, Barbara. You need a champion. You actually need two champions, one on both sides of the aisle. You need a clinical safety professional, and you need a safety data scientist working together, because it takes both in partnership to deliver these ideas. And one barrier that we need to overcome is a lot of leaders and decision makers in companies have an efficacy mindset. They don't have a safety mindset yet. So they're thinking about this really in terms of p-values, right? Nominal, unadjusted p-values, and it is likely to find something, even if it's a false signal. And we need to communicate to them that we have these sophisticated methods that takes into the context of the data and helps minimize the false positives. So using these methods can not only help identify signals, it can also help authoritatively refute signals.
Yeah, thank you, Greg. And I think the other thing people say to me is that sometimes, people outside of safety at the company are very concerned by, uncovering safety concerns that, might not turn out to be anything really impactful in the long run, but raise, issues with the product during development that become problematic. But I think that, the earlier you can pick up signals and then evaluate them and really try to understand them, and if there is something there of concern, try to see if you can figure out ways to manage that risk. So the product, if it is looking efficacious and you do think it has a positive benefit risk assessment, to find ways to be able to move that product still along in development, still keeping in mind how to minimize patient risk. Lian, you have your hand up too?
Yeah, so I just want to highlight how we can implement this procedure. So it's very important to embed that to the sponsors safety governance procedures, because once we did this practice, there was safety signal will be highlighted. So which body will be review those data? So that need to pre-planning and engage with leadership teams, so that we can pre-planning and also have set up the pilot, set up the review bodies, So in large sense, the aggregate safety assessment planning process is really important to help us to align with multidisciplinary teams, and also planning how we look at the data and the feasibility of the data, how to implement, how to review, how that impact the clinical development. All those things can be putting together.
Thank you. Shihua, you have your hand up as well?
Yeah. So I think, I mean, I probably want to echo some of the presenters' comments as well. And I think we need to select a battle to fight, that battleground to fight. Everybody understands safety is important, but we don't necessarily to really use a very a very stringent safety monitoring in all the program or in all the studies. So we need to really select the battleground to fight, and using using those safety methods in the real the programs that really need to detect that. For example, I'm working on the rare event field. Sometimes if only one event could actually already raise some kind of safety kind of concern. So in that case, we probably don't need a very sophisticated safety monitoring. But there, for some other safety events, we probably want to really implement an upfront safety monitoring plan, and that also need a lot of discussion between the team, including statistician, probably study manager, the clinical safety experts, etc. So we want to have the plan as concrete as possible, because I mean, we talk about blinded review, we talk about unblinded review. We need to really make them specified, and everybody agree. Yeah, thank you.
Thank you. Nils, you have your hand up?
Yeah. Just a side note, something that helped us at UCB was to offer a mix of technical and non-technical trainings for aimed at different populations, from clinical colleagues to statisticians and programmers, and this way before the method was completely finalized and way before it became a process or an SOP at UCB, and that was really helpful to soften the blow of adopting something really new that sometimes a bit scary.
Yeah, thank you. Well, maybe we can move on to the second question. I mean, we've kind of touched on parts of this question, I think, already, but perhaps people can share any practical solutions to implementation of these quantitative methods and safety monitoring. I think, looking at the ones that are listed here on the slide, I do think if you use the Bayesian methods and need to incorporate an estimate of background risk for anticipated events, that can be challenging in my experience. Not all companies have epidemiologists to help. And even so, I think that, a lot of times the epidemiologists, that their work doesn't really, involve this type of activity for estimating background risk for rates for clinical trials. And so I found that sometimes it really did fall on the safety group to try to help with that. But, it is really challenging. And looking at public sources of information, our ASAP task force has been looking at that. And you can see that the quality and the transparency of the safety data from clinical trials that's available in public sources is quite highly variable. And so you really have to piece together almost like a jigsaw puzzle, background rates for, anticipated events in different disease indications that it might be being evaluated from various public sources. You can often kind of do it, but it's very time consuming and it's challenging. And so I hope that, we're writing a paper right now kind of addressing some of this, and hopefully we can make some recommendations. But I just wonder if anybody else wants to comment on, any of these bulleted challenges and implementation and how they've been tackling them at their organizations. Brian?
Yes. So, as you were talking about the estimating the background risk, one of the things about looking at blinded data is that you can, for an ongoing trials, you can really start understanding some of the risks of your assumptions. So I have an experience in which the background rate was actually an inappropriate background rate. But it was how we sort of framed our sort of thinking around this particular endpoint. By seeing it early, right? All of a sudden we saw this elevated risk. We were able to rethink and reinterrogate, well, what were those numbers and what should we be expecting? And for our operationalizing our methodology, doing additional sensitivity analysis around the background rates. So, background rates are extremely tricky, and you should realize that just because you see something, you have to look at all the different factors that go into a methodology. So that is a lesson that I wanted to share with the group.
Yeah, thanks, Brian. I mean, Brian and I worked on a large program together where I think that, we used BDRIBS as one of the methodologies. And I do think it was highly successful at identifying the safety risks that came up later during the submission process. And so I think that, with large clinical trials, this can be very valuable to apply these types of methodologies. So, Lian, you had your hand up as well?
Yes, so I completely agree. This is tough questions. So to have this quantitative framework works, we have to have a good background rate estimated. So in previous presentation, you may notice that we will just assume that we have the background rate, but in reality we don't. So exceptly, Barbara, you just mentioned using the public data. So another approach I think we can use is using the patient level data. We can either get from the real world data, or we can get from the existing clinical trial data. So I just want to advertise that we have identified this issue many years ago, and working stream 1 and working 3 is collaborating to tackle these questions. So so I think we can using the currently the large data set to get it estimated, and in addition to that, it also has to be to pre-planned. So aggressive evaluation process can be also to leverage it to have a list pre-specified, including the approach, the data source, to estimate the background rate, so that it can be appropriately plugging into the quantitative framework to have this method works.
Yeah, thanks, Lian. I mean, I think the real world data is certainly an option. For smaller companies, they don't have access to those kind of databases, which often, require significant monies to have access. Or otherwise, I think they have to collaborate with external collaborators. The other issue, obviously, is that the data is coded in ICD 10 codes, for example, whereas clinical trial data is coded in MEDRA. And also, there's just a lot of differences between real world data and clinical trial data. And my personal experience is that the rates coming out of real world data almost always are significantly higher than what people actually see in clinical trials, which might reflect differences in the patient populations. But certainly, if you have no other, source of background data, then real world data becomes a very valuable potential source, just keeping in mind all the caveats to that and trying to get an apples to apples comparison as much as you can. But yeah, so I think that's a great point. Saurabh, you have your hand out again.
Yes, thanks, Barbara. So I just wanted to follow up on what Brian and you have mentioned. The transparency of the process is very important, as you mentioned. Estimating background rate has two components. One is the point estimate, another is the uncertainty around it. So when we bring new data or external data to estimate that, we need to be mindful about the uncertainty that comes with it. For example, BDRIBS method provides that framework where we have a prior that kind of builds the uncertainty around the modeling and around the inference. So I just wanted to make sure that we keep that in mind, that sometimes there are uncertainty around the rates we see, but when we use the trials, that uncertainty can be handled in a very quantitatively rigorous manner.
Yeah. Dooti, do you had comment on this one?
Yes, I just wanted to follow up on one comment you made, Barbara, and also some of the other responders. This was about choosing your battle, right? We talked about we shouldn't really focus everywhere, and we really need to pick and choose which programs will benefit the most. And in my experience, what I've seen, the programs which are having that kind of high priority are also some of the busiest. The teams are always running, and therefore it is harder therefore to implement something completely new and say, here I come with my new fancy method. I can assure you it will provide value. So I feel that what is really helpful is to start that conversation earlier on, and not just about what method to use, what data to use, but also do you have the right infrastructure and the scalability you need to implement this. So speed and customization are probably two very important aspects to also consider. And this is a little bit going back to thinking on pre-planning. In order to implement a certain approach, what do I need? Do I need the infrastructure? Do I need to upskill some colleagues? Do I need to start that conversation earlier on so that we can actually be successful when the time comes? So just wanted to bring that point. Thank you.
Yeah, and I think our ASAP working group has published about the aggregate safety assessment plan template and its use in safety planning, and that, even we call it the safety topics of interest table, but that table I think is even really useful even in phase 1. But we've said that, starting this systematic planning for aggregate assessment, certainly by phase 2 is really important. And I think that lays the groundwork oftentimes for using these more quantitative methods. So Cindy?
Hi, Barbara. I was just going to add to some of the points and give an example that we ran into, how it is very challenging to get data. Can you hear me okay? Oh, I'm sorry. To get the data, that is definitely a challenge to access the data and also curate the data in a way that it's usable is a big challenge. But sometimes even when you believe that you've got the background data, you've pulled information from the literature, you've analyzed it, and you've produced your analysis and you think you've done well, sometimes you haven't. And that gets back to that point I think Nils was making earlier, really understanding your patient population. And if you don't have a clear understanding of some of the patient population, particularly in the earlier phases of development, you might find that even when you think you've got it defined, you don't. And when you have something that's escalated unblinding, and you think there's a big problem with the blinded data, but when you get to unblinding there isn't, then that helps you understand that maybe you didn't have your patient population right. So that was an experience we had in one application, particularly when it was when we were using external data sources solely. Or literature, and the data is very inconsistent as well. So some of that population information, if you're having to pull from the literature that isn't consistently available, can have a big impact on your background event rate. So just thought I would share that. But other than, but do definitely support the planning aspect to what Dooti said, that we have found that in the absence of that, it's very difficult, and to have some written procedures for how you would implement this. And I think one of the speakers presented, Lin, said what you're going to do with the information once you have it is very important, because I think teams are a lot less reluctant to take on the methodology if they don't know how they're going to action the outcome.
Yeah. I think at least with FDA, my understanding with their draft guidances, like the 2021 draft guidance on IND safety reporting, is they're really looking for companies to identify earlier in development anything that would have ultimately ended up in the warning section of your label. So they really don't want those things to be total surprises necessarily at the time of submission, but to be identified earlier. And that way too, again, if the product does have good efficacy, maybe there's a way to manage that risk so that you can still have a positive benefit risk for that product that would allow it to be approved and used for that indication. Shihua?
Yes, I think I also fully agree with what the previous speaker saying. And not only try to choose the battle for the fight, and also we need to really actually, I mean, when we fight, we need to fight well. And in order to do that, we really need a lot of effort for this kind of pre-planning. And I think we talk a lot of methods, but I also want to say we shouldn't really underestimate a lot of operational work as well. I mean, we talk about okay, you need to have this kind of a plan on okay, which method to use, what data, which risk, and which kind of parameter you need to set in order to do this appropriate safety monitoring. We also need to really pay attention to the communication plan as well. For example, what if we see some signal? I mean, who should we actually talk to? And as I mentioned, blinded, unblinded review. If this is blinded review, probably it's easier. But if this is unblinded review, how can we actually set up the firewall? And I think the communication plan also very important. So I think there's really a lot of considerations from methodology-wise and also operational-wise to think of. So the upfront pre-work is really actually important, and we shouldn't really underestimate the amount of work. Yeah.
That's why I think if you can get the safety management team or whatever your company calls that to be discussing some of this during phase 2 before they get crazy busy, as Dooti mentioned, for some of these big programs in phase 3, and then they're so busy they don't want to, listen to something that might create more work that's not been standard up to that point for them. But I think that, really safety planning earlier on, as you are anticipating going into phase 3, so before you ever get to phase 3, is really critical. Or oncology earlier, if you're generating a lot of serious adverse events, even in earlier trials, I think you need to be really focused on this earlier as well.
Yeah.
So is there anybody else who wants to comment? Especially, I don't think anybody's necessarily touched on deciding a number of looks of aggregate reports. If anybody wants to speak to that at all, the third bullet there.
Maybe I can just one more point related with actually the number of look as well, which is actually related with data. So I mean, in order to really support a good safety monitoring, we need data. I mean, if you really think like either continuous looking or the sequential looking, we need the data actually enter into the Shining app, and then we can see what kind of graphs they generated, so that we know it's cross-boundary or not cross-boundary. So I think the data cleaning is also very important. We certainly do not want, there are some kind of unclean data, which really creates some kind of hassle regarding the safety signal. Yeah. So I think the data cleaning and also the data plan for the transfer, that's also important. Yeah. I'm going to stop here. Thank you.
Okay. Thank you. Lian, you have your hand up?
Yeah, just quickly, I think the continuous look of data, they have better statistical properties, but when we implement, it's really need to consider operation issues, just like the previous speaker just said. So especially the data cleaning and also when the data availability is it cannot be continuous a lot of times. In addition to that, we can think about how valuable to have a continuous look, because the sponsors safety governance bodies, usually they have scheduled meetings, look at evaluate safeties. So how much value that we do, it takes so much effort to do it continuously, but we only look at sequentially.
Thank you. Brian?
Yeah, so a kind of a practical aspect, which actually goes back to what Shihua was mentioning, is that you know often we've used these things around the same time as DMC looks, right? So the team is already preparing data for another purpose. So piggybacking on that sort of already planned output is one thing that we can do. Though I also have had the situation where by doing that, you know, increasing, if you want to look at the data more often than what's being done for the DMC, you need to be very thoughtful about how that data is equally important from a cleaning perspective, from a, you know, what queries are out there. You know, it doesn't have to be a pristine like a database lock, but you do have to understand what sort of information is out there, because that can lead to some sort of extra variability on your conclusions. So
yeah, and if you're using adjudicated data, I think for rates, then you definitely, like Brian says, you do have to keep in mind, like, what's the status of your adjudication of events at the time that you're looking at the data. Saurabh, you have your hand up?
Yes. So one other component is that how much data will add any meaningful information. So we can actually plan ahead of time and see that if we need to wait for every 50% years or 100% years, would that be a meaningful information to add to the existing knowledge? And that can help planning also. Just a quick point.
Okay. Anybody else have anything they want to add? Or anybody in the participants on the call that had questions or wanted to add anything from their experience? Raise your hand, anybody? No? I mean, I just would say in conclusion that, you know, that it is doable. It does take a bit of work the first couple of times you try to implement these methodologies, but I think once you've done it a couple of times, it gets easier, for sure. It gets easier, but every program has its own, you know, challenges that might be unique to it. So, you know, you do have to tackle those each time, you know, you're starting in with a new program. But I think that, you know, for larger programs or programs that have a large amount of serious adverse events, I think these quantitative methods can add a lot of value. And but it is really important for the safety personnel and the stats group to collaborate closely together, both for championing the use of the events, for prioritizing which programs these methodologies would be most useful for, and then and how to execute on it, and then how to handle the communication pathways between, you know, the unblinded assessment entities, you know, that might be looking at the data if you're using the trigger methods, or the data monitoring committee if that's running in parallel, your safety management teams, and then your safety, you know, senior personnel in the company, you know, that you may have to communicate to as well. So yeah, I think those are all things important, and training, and, you know, just having everybody really understand what you're doing and why you're doing it. So I don't know, Rima, if you have anything else you want to add here at the end.
No, thank you, Barbara. This was a great discussion, and I was taking notes, and I'm so glad that we'll have also the recording for this, for all the speaker presentations, for all the references for their methods, and then also, as some of them have already pointed out in the chat, they're all available for questions if you have any questions. I wanted to leave with a big thank you for all the participants, but also for a call to actions for those of you who are still on the line. That is, we did our best to try to collect all the methods that were developed or used, especially the ones that have tools that are available or are in development. But if we missed anything that is important that you're using within your organization or that you would recommend, that you found useful, please contact me, and we'll happy to add that as a method or tools in the list of references that were not discussed today. Also, if you are interested in learning more about some of these methods that were presented today in just 5 minutes, please email us also, because that can make you a participant in shaping the future of safety webinars that are organized either by ASA Biopharm or some of the sister organizations. Also, if you're interested in development new methods, please join a professional organization that discusses safety methodologies. That could be ASA Biopharm, that could be Fuse, or that could be PSI. These are really forums for us not only to share common experiences and problems, but also to work together across different companies to develop methods that may have a bigger weight than if you're sort of just advocating within your own organization. So again, thank you very much for everyone, for all the participants. I found this fantastic. I can't wait to even rewatch the video, and I hope that this is only the first one of the 2025 and that there will be other follow-up webinars later this year. Thank you so much.
Thanks for pulling this all together, Rima. This was a great landscape. I would like to see more webinars like this.
Yes, thank you. It's great.
Take care, you guys. Bye-bye.